I'm Daniel Levine, and this is the Bio Report. Epigenetics, the layer of chemical switches that control how genes are turned on and off, can act like cancer's operating system when a single epigenetic enzyme becomes essential for tumor survival. K36 Therapeutics is developing first-in-class medicines that block an epigenetic enzyme that helps certain multiple myeloma cells grow, change identity to escape treatment, and become resistant to today's drugs. Terry Connolly, CEO of K36, discusses a new way to fight cancer by changing how cancer cells read their DNA instead of chasing one mutation at a time, how K36's experimental therapies aim to resensitize tumors to existing treatments, and the potential to create new options for people whose cancers have stopped responding. Terry, thanks for joining us.
Oh, my pleasure to be here today, Dan. Thank you for hosting me.
We're going to talk about epigenetics, cancer, and K36's efforts to develop treatments for hematological and solid tumors by targeting an epigenetic driver of certain cancers. Before we dive into that and what you're doing, I thought it would be useful to remind listeners what we mean by epigenetics.
Certainly. So epigenetics refers to the layer of regulation that controls how genes are turned on and off without changing the underlying DNA sequence. If you think of DNA as hardware, epigenetics is more like the software, where we call it reprogramming, that tells the cell what programs to run. It determines cell identity, how cells respond to signals, and how they benefit or how they behave in different environments. This regulation happens through chemical modifications to DNA and to histone proteins, which package the DNA. These marks act like switches or dimmers, turning gene expression up or down. In cancer, that system becomes dysregulated. Instead of normal cell growth, you get widespread changes in gene expression that can drive tumor growth, survival, and resistance to therapies. What's important is that, unlike genetic mutations, epigenetic changes are reversible, which makes them very attractive therapeutic targets. That's where our work comes in. We're targeting one of these key epigenetic regulators, NSD2, that plays a central role in driving certain cancers.
NSD2 is an epigenetic enzyme. What exactly is its role, and how can aberrant NSD2 activity drive malignancy, therapy evasion, and cancer cell survival?
NSD2 is an epigenetic enzyme that controls how genes are turned on and off by modifying chromatin, specifically through a mark called H3 K36 dimethyl. In cancer, that system gets dysregulated. In multiple myeloma, for example, a translocation drives overexpression of NSD2, and in other settings, you see activating mutations. The result is the same. You get widespread reprogramming of gene expression. What makes NSD2 important is that it doesn't just affect one pathway. It reshapes the entire transcriptional landscape in a way that helps cancer cells survive. It promotes things like adhesion to the microenvironment, resistance to therapies, and even the ability of cancer cells to change identity and evade treatment. The tumor becomes dependent on this altered epigenetic state, and this dependency is the vulnerability that we exploit with our drug KTX-1001. When we inhibit NSD2, we're essentially reversing that programming. We reduce the methylation signal, disrupt those survival pathways, make the cancer cells more vulnerable. It's a different approach. We're not just trying to kill the cells directly, we're reprogramming them out of a malignant state, which we think can lead to more durable responses, especially in combination with other therapies.
In the case of the cancers you're pursuing, is NSD2 the driver of these cancers, or Is something else giving rise to them and then it's exploiting this?
That it can actually be both, and that's what makes this target so interesting. So, in the case of a subset of myeloma patients called T414, NSD2 is a founding driver. That T414 refers to a genetic translocation where chromosomes 4 and 14 swap genetic material, and the translocation puts the NSD2 gene under the influence of a strong enhancer. So, very early on, it's overexpressed and is actively shaping the biology of the disease. But in other settings, NSD2 plays a role, more of an enabling role. The cancer may arise through other genetic events, and then NSD2 becomes upregulated or activated later, helping the tumor adapt, survive therapies, or become more aggressive. So you can think of NSD2 as both a driver and a dependency. In some cases, it helps initiate the disease, and in others, it reinforces and sustains it, particularly under treatment pressures. What's consistent across both scenarios, however, is that once NSD2 is active, the cancer becomes reliant on that epigenetic state, which is exactly what we're targeting.
Your lead experimental therapy, this is KTX 1001. This was discovered by Novartis. How did K36 come to license the asset?
The opportunity to license the molecule we now call KTX 1001 emerged during the early days of the COVID pandemic in late 2020. At that time, Novartis made a strategic decision to convert one of their sites from a discovery center of excellence to a development center of excellence. So any drug candidates that were not in clinical testing became available for out-licensing. And the molecule we now call 1001, the NSD2 inhibitor, was one of those programs. Our scientific founders immediately recognized the significance of the biology. NSD2 had long been understood as a key driver in multiple myeloma, but it had been difficult to drug. Seeing a credible small molecule against that target created a very compelling opportunity. For me, personally, stepping in as employee number one, it was a thrilling opportunity. At an earlier stage of my career, I worked at a company called Celgene, a company that had a very large commercial presence in multiple myeloma. And it was at Celgene that I first learned about NSD2 and its importance in multiple myeloma. I later worked in business development for that company, and this was a target that I continued to follow very closely and believed very strongly in. So when I learned that our investors were starting a company targeting NSD2 with a candidate from Novartis, it was one of those rare situations where the biology of the molecule and the timing all aligned for me.
NSD2 had long been considered undruggable. What convinced you that KTX 1001 was worth building an entire company around?
Yeah, so it had been considered undruggable because of the chemistry, not the biology. The biology was very clear and compelling. When Novartis demonstrated you could, in fact, drug this target with a selective small molecule, that was the key inflection point for company build. And the decision to build a company around the molecule came down to a few things that are really important for drug developers to consider. First, the NSD2 biology is compelling and strong. There were more than 25 years of biology demonstrating that this pathway is important to cancers. NSD2 is not a peripheral pathway, it's a core epigenetic driver. Second, there's a testable clinical hypothesis. There's an identifiable patient population with unmet medical needs, and testing the 414 translocation is part of their standard of care. So we essentially knew the patients that we'd want to test our drug in. And the presence of this translocation has historically been considered a high-risk feature, and patients with this translocation did not have the same benefit as standard risk patients. Third, there are biomarkers that we can measure during early clinical development to demonstrate that we're inhibiting the target. So early in development, we gain confirmation that we're achieving the same biology in patients that we achieved in the lab. Most importantly, the emergence of a credible molecule with the right properties to take into clinical development had been identified. 1001 showed selective inhibition was achievable, and there was compelling preclinical evidence that demonstrated modulation of this target could translate into meaningful biological effects. And finally, it provided an opportunity for me to build a company and a team around the molecule. So with the right chemistry, the right CMC, the right clinical strategy, we believed we could move efficiently towards proof of concept. So it wasn't just a belief in the molecule, it was really a convergence of validated biology, enabling chemistry, and execution capability that made it worth building the company around.
Your lead indication is multiple myeloma. This is an enzyme that's overexpressed in about 20% of cases. Are you using a companion diagnostic to identify patients most likely to benefit?
Patient selection is a key part of our strategy, particularly as we move beyond the dose escalation phase of our study. In multiple myeloma, NSD2 is most clearly driven by the 414 translocation, so we're prioritizing that genetically defined population where the biology is strongest. As we move into the dose expansion beyond monotherapy, we're using a central lab to confirm T414 status to identify eligible patients. At the same time, we know NSD2 overexpression extends beyond 414 patients. There's a broader group of patients with what we call NSD2 high biology, and that's an area we're actively exploring. We've examined a public data set and have identified that there's a subset of patients who lack the 414 translocation but have high NSD2, and their clinical outcomes are comparable to T414 patients, and significantly worse than those who don't have the high NSD2. We're actively working on a development assay to identify those patients. But for today, identification of the 414 patients is our primary approach. But longer term, we do see an opportunity to expand into the broader biomarker-defined population as the data matures, redefine diagnostic strategies.
And are you looking at this as a monotherapy, or are you looking at it in combination with other potential therapies?
We've completed the monotherapy dose escalation, which demonstrated safety and characterized the pharmacokinetic and pharmacodynamic properties of the molecule. That confirmed for us that we're hitting the biology in patients, we're seeing the expected epigenetic effects, and we have a tolerable profile to build on. From here, the focus is very much on combinations, as multiple myeloma treatment is dominated by combination therapies. And because NSD2 inhibition works by reprogramming the cancer cells, it's particularly well suited to combination approaches. NSD2 is used by cancer cells to develop resistance to therapies. So one of the ideas is that in combination, you prevent the resistance so that patients will be able to benefit from a particular therapy for a longer period of time. So the idea here is that we reset the tumor biology, restore or enhance sensitivity to existing therapies. So we've generated very strong preclinical data showing synergy with multiple standard of care agents, including proteasome inhibitors, IMiDs like pomalidomide, and CELMoDs like mezigdomide. And we're now advancing combination cohorts of those agents into the clinic. So monotherapy is an important first step in drug development, but the real opportunity and where we believe we can drive the most meaningful and durable benefits for patients is in combination regimens.
What's known about it from the studies you've done to date in terms of its safety and efficacy?
So we've fully characterized the monotherapy dose escalation. We have demonstrated target engagement, wide therapeutic range that to explore dose optimization, and preclinically we've shown the ability to combine with a number of agents.
What's the development path forward?
The development path forward is really focused on moving into earlier lines of therapy and advancing combination regimens in this biomarker-defined patient population.
Bristol Myers Squibb is an investor, and you also have a clinical collaboration with the company. What's the nature of that relationship, and what are you working on together?
They are an investor, and we do have a clinical collaboration. They participated in our financing, which reflects their belief in the NSD2 biology and the potential for our molecule to work with their drugs. Beyond that, we have an active clinical supply collaboration for mezigdomide, one of their multiple myeloma agents that's in late-stage clinical testing. We have preclinical data that demonstrates strong synergy between mezigdomide and KTX-1001, and that collaboration allows us to explore that combination in the clinic before that agent becomes a commercial stage or commercially available asset. More broadly, the relationship gives us the ability to align with leaders in myeloma therapy as we think about future development paths and potential registration strategies. We're a small company, so it allows us to leverage the thoughts and the capabilities of a larger company. So it's really both strategic and highly practical, enabling us to move faster on the combinations that matter most for patients.
How are you thinking about other potential collaborations, and where do you see your partnering strategy fitting into your pipeline?
In the multiple myeloma program, we continue to think about partnerships very strategically, always through the lens of what will most efficiently translate our science into meaningful patient benefit. First, we look for strong mechanistic rationale. NSD2 inhibition is highly complementary to existing therapies. So we're focused on partners whose agents can combine in a way that enhance or restore sensitivity and drives deeper responses for patients. Second, clinical execution matters. The right partner brings not just an asset, but real expertise in development, access to patients, the ability to move quickly in well-defined populations, connections to key opinion leaders in the field. Third, we value alignment on a long-term vision. We're building towards a precision medicine approach anchored in NSD2 biology. So we look for partners who see the opportunity to develop combinations in a thoughtful, data-driven way, potentially into earlier lines and registrational settings. The BMS collaboration is a good example of that model. And going forward, we're open to additional partnerships that fit those criteria, whether in hematological malignancies or as we expand into to solid tumors.
You do have a second NSD2 inhibitor in development. This is KTX 2001. This is in development for metastatic castration-resistant prostate cancer. Where did KTX 2001 come from? Was this discovered in-house? Did you in-license this?
We did not in-license this. This is an internally developed program. We think of it as a next-generation NSD2 inhibitor that's really built on everything that we've learned from 1001. So as we advanced 1001 in multiple myeloma, it became clear that the biology of NSD2 extends beyond hematologic malignancies, particularly into solid tumors like prostate cancer, where NSD2 is linked to something called lineage plasticity. That's this change in cellular state that allows the cells to develop resistance to therapies like androgen receptor pathway inhibitors that are used in metastatic castration-resistant prostate cancer. So 2001 was designed specifically with that setting in mind. We optimized the molecule for the pharmacologic and biologic requirements of solid tumors, including properties like exposure and penetration into solid tumors. So while 1001 allowed us to establish clinical proof of concept in a genetically defined myeloma population, 2001 represents the next step, extending the NSD2 platform into larger indications like metastatic castration-resistant prostate cancer. It's really a deliberate portfolio strategy. Start with a highly validated, um, well-defined patient population, and then move into a broader development around the same biology core.
Now, what's known about it from the studies you've done to date?
Early days for that program. We've just started the clinical testing, but so far the data supports why we built it. Preclinically, it shows the potency, selectivity, and profile we wanted for solid tumors. What really drove the program is the growing evidence that NSD2 contributes to this lineage plasticity and treatment resistance in advanced prostate cancer. That gave us the rationale to move into mCRPC, and the program is now in first-in-human phase 1 study where we'll explore monotherapy and in combination with darolutamide, another drug we have access to via supply agreement, in this case with Bayer. At this stage of clinical development, we're focused on safety, PK, PD, and early activity, and in both monotherapy and the combination. Biology is really compelling, and we have a very comprehensive translational science program that aims to verify that in patients we will see the same biology changes that were observed in the lab.
And what's the development path forward for 2001?
So we're in metastatic castration-resistant prostate cancer now. As I said, where the focus is on clinical safety, evaluating the properties of the molecule like pharmacokinetics and pharmacodynamics, as well as early signals of activity, both alone and in combination with darolutamide. Where it gets particularly interesting is the biology of NSD2 is linked to this lineage plasticity, which is a driver of resistance in advanced prostate cancer. And it seems to really be upregulated in patients that have something called treatment emergent neuroendocrine disease, or TNEPC. And this is an area with very high unmet medical need, no approved therapies to date, really an aggressive form of prostate cancer. So our strategy is to move quickly into combinations with darolutamide, where we think we can potentially prevent or resist that shift and restore sensitivity, or have patients remain um, responsive to AR pathway inhibitors. And as the data matures, we'll expand into selected patient populations and refine where the signal is strongest. Longer term, the goal is to move to earlier lines of the disease and build towards a registrational path in a biomarker-informed setting, including patients with features of aggressive variants like treatment-emergent neuroendocrine prostate cancer.
As you think about the cancer landscape, how broad a potential is there for this type of approach?
Epigenetic dysregulation is a hallmark of cancer, but what matters is when a specific regulator becomes a true dependency. For NSD2, we see three primary ways where it becomes elevated. The first is in multiple myeloma, where this genetic 414 translocation drives that overexpression of the protein, and it's a founding driver in that subset. The second is this lineage plasticity hypothesis that we're exploring in prostate cancer, where cancer cells upregulated these pathways to resist therapies. And it's very well described in this subset, but it's also been shown to occur in lung cancer and a range of other solid tumors. And the final is there's a gain of function mutation in a specific part of the NSD2 protein where you don't overexpress the protein, but you increase the catalytic activity of the enzyme. So the net effect is that you make more of this K36 dimethyl with the same amount of enzyme, and essentially you net out at the same biology as you would if you overexpress the protein. And in this gain of function mutation is seen in a number of hematological malignancies like mantle cell lymphoma and also TALL. So NSD2 acts as a driver and an enabler depending on the disease context. I think from a development standpoint, that creates a very broad opportunity, but we're focused right now on where the biology is strongest and expanding in areas like prostate cancer and eventually into other areas like mantle cell.
And you talked about the differences between 1001 and 2001, but how broadly applicable might they be in other hematological or solid tumors?
The therapies are highly tailored, but the biology gives us the potential to expand. So we're starting in this very specific, well-defined patient population like 414 myeloma, where NSD2 is a clear driver. And there, that's where we can be the most precise and demonstrate the strongest clinical signal. Beyond that, there will be an opportunity to expand into combination therapies. We see it elevated through multiple mechanisms, the translocation of myeloma, this gain of function mutation in other hematological malignancies, lineage plasticity in prostate cancer, and even in our own data, we've shown that it's elevated in multiple myeloma patients that don't have the translocation. So I believe that this is a pathway that cancer finds, it turns on, and it very quickly realizes that with upregulation of this pathway, there's a benefit for it. And I think that we're just scratching the surface of NSD2 biology. And eventually this will become a very important driver across a range of indications.
K36 has been successful at raising money during a difficult financing period for biotechs. How far will existing cash take you?
Yes, we have been fortunate. I think one of the things that was made it a little easier for us when we raised our B round is that we had real assets. It wasn't a platform where we were hoping to discover a molecule. We were in the clinic at the time, and good assets continue to attract capital. So we've been thoughtful about how we finance the company, how we build the company, and especially in an environment where raising capital is still challenging, although that seems to be turning now. To date, we've raised approximately $100 million. We've been very disciplined in how we deploy that capital, really focusing on trying to put as much of that money into generating meaningful clinical data to basically derive a signal that will show that our drugs benefit patients, and we'll continue to build on that. Current capital takes us through the key inflection points over the next year and a half. That includes advancing 1001 into 3 combination cohorts for multiple myeloma, and then completing the dose escalation with 2001 in prostate cancer and the combination of 2001 with darolutamide.
And as the environment has warmed a bit to biotech, have you noticed any shift in the nature of the conversations you've had with investors?
We've had more inbound interest. So investors have been reaching out to us, asking us about our plans to raise capital. And there's certainly several who have said that they would like to be on our call list when we do decide to raise the next round. But the investor conversations are very disciplined. I think over the next 12 to 18 months, we'll be looking to raise additional capital. I think the bar for us remains high. Investors are focused on clear clinical validation, differentiation, and capital efficiency. One part that is, a bit of nuance for us is that being first in class can be a double-edged sword. It's exciting, it's thrilling to be the first to explore the biology, but it also can sometimes make investors a little more conservative because you're defining a new space and a new path rather than following a proven path where you're saying that you've got a molecule that's differentiated from the one that's first, and that somehow you're going to be the first to be second, and that's your story. That said, we've seen a high level of engagement from a lot of support from our existing investors, and growing interest from the broader community as well. I think there's a lot of recognition that targeting epigenetic drivers like NSD2 goes after the core programming of cancer in a fundamentally different way. And there's certainly, a bit of a rise in the epigenetic space right now with what companies like ORIC and Pfizer are doing with their phase 3 studies in prostate cancer with EZH2, another lysine methyltransferase.
Terry Connolly, President and CEO of K36. Terry, thanks so much for your time today.
Dan, thanks for finding K36 and listening to our story and promoting epigenetics. It's a pleasure speaking to you this afternoon.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.